Good afternoon, my name is Trip Cooper and I was a part of a group that maintained and conducted research on a hydroponic NFT system that contained 2 varieties of arugula and 2 varieties of kale. Weekly, we recorded information on the system that will be shown below and did our best to properly maintain our system. To begin, our hydroponic system was greenhouse grown, which allowed for a controlled growing environment. We could monitor the air temperature, control the solution temperature, and carefully monitor each step of the growing process. To provide a basic overview, an NFT system is basically a dark channel at a downward angle that allows the solution to flow with gravity. The water flow is constant due to a pump pushing the solution. The roots of the plants are also not completely submerged, which allows for the solution to flow more freely. On top of this, air filtration goes through for a faster growing period with more harvest.
So now I'm going to be talking about when we did our plugs. So you can see in our left photo there, the plugs look really pretty and really just bushy and full. But then we would break all of those apart into smaller individual single plants and then put them into the holes of our NFT pipes. So in the middle photo there, you can see where we had finished filling all the holes, and it definitely looks very scarce and not as great, but it worked out. So now we have our height measurements. So for this, we took our heights each week, and then I took every single one of those data points from each week from each treatment and averaged them all together, and that's what you'll see in the graphs and data table there. So you can definitely tell that during the first week everything was normal and uniform, and then during the other weeks, treatment 1 definitely had some issues there. You can see that it barely had any incline of growth, while the others had pretty big changes of growth, while treatment 1 barely got there. Yeah, we had a lot of issues with this one treatment, and it just really never got to the height of all the other treatments. So with width, we took north to south measurements as well as east to west, and then I just put those all together and averaged them as well. And for this one, you can definitely see that treatment 1 did have higher width than it did height, but during week 2, it somehow did decrease a little bit for treatment 1, but luckily it got back up there a little bit and got close to almost 7 inches during week 4 for treatment 1, while the others had very gradual width as well as a lot higher and better. So you can definitely see that the anthocyanin levels here were kind of all over the place, especially for treatment 1. You can see that during week 1 and week 2, treatment 1 was going, it was pretty high and had a little increase, but then randomly during week 3 just plummeted down. And then increased even higher during week 4. So it's a little strange. That treatment definitely, you really can see that it was troublesome and there was a lot of issues with it. So your chlorophyll levels are definitely a lot more uniform and nicer than your anthocyanins, where there isn't too much differentiation for the first 2 weeks. During week 3 and 4, you see a little incline where treatment 2 was actually lower, and then during week 4, treatment 1 ended up being higher than all of them. So it was definitely a lot better levels than we did for anthocyanins. So our pH was actually probably one of the things that didn't go completely wrong. We had so many issues with our water systems, but we somehow kept pulling in some pretty great basic pHs. So that at least is nice. You can see definitely a lot more consistent, uniform, a little change, increase and decrease during week 3 and 4, but other than that, it's not as drastic as a lot of the other weeks or a lot of the other measurements. So our dissolved oxygen was also another one that was kind of a little more uniform, consistent, not as much as our pH was, because you can definitely see that it was kind of high during week 1, and then all of them kind of decreased during week 2 and 3, and then all 3 treatments had a pretty high dissolved oxygen during week 4, but they're all pretty close together during week 4, so that's pretty nice. So EC, our electrical conductivity, there was a lot of fluctuations on this graph. So you can see on week 1, treatment 1 was definitely pretty high. Then you had week 2, a little decrease for treatment 1, and then week 3, it just extreme decrease, went all the way to the bottom. It was a little strange, and then a extreme increase for treatment 3 during week 3. Very strange. And then during week 4, treatment 1 came back, increased even higher than before, and treatment 3 just went all the way to the bottom. So here is some data comparisons from our different harvest. So harvest 1, especially for the plant heights at the top, was a lot more consistent. You had a better gradual incline than you did for harvest 2. You can definitely see there that during week 4 for harvest 1, they got pretty tall, pretty big, but then week 4 for harvest 2 barely even got there. It was pretty sad. You can see that there was a lot more issues during harvest 2 than there was harvest 1. And then for EC, you can see here that for harvest 1, definitely a little not as fluctuated as it was for harvest 2. Harvest 2, it was definitely everywhere over the board. You had random increase and decrease, while harvest 1 had some slight increases and slight decreases, but not as drastic as harvest 2. Then our anthocyanin levels, so for harvest 2, treatment 1 was insane with its anthocyanin levels. It was a lot more gradual and consistent in harvest 1 than it was for harvest 2.
Hi, my name is Ross McKinstry, and I'll be talking about the problems that we came across in this experiment. One were the diseases and the stress that were being put onto the plants throughout the weeks before harvest. Within the first couple weeks, as the leaves started to develop, we could see chlorosis. As you can see in the top right, this isn't our kale, but this is an example. But what we had were the tops of the leaves were just yellowing. So that was one thing we encountered. That was like within the first 2 weeks. The second thing we ran into was that the first treatment was not growing as well as the second and third treatments. The first treatment was getting scorched and it was stressed, making it hard for that treatment to mature and start growing at a faster rate like the second and third. And then a really weird thing that we came across was that some of the flowers in the first and second treatment had started to have white flower blooms at the top. We figured out that it was from stress, I guess from not getting enough substrate, or just from it being clogged and there's not enough airflow through it. But yeah, I mean, that was one thing that was pretty cool that happened. Second, pests. This was a major issue. They were eating our roots. They were all over the treatments. You get on top of the leaves and you look down and you have these little red mites right here, all over your clothes. Then we found that in the roots, in the channels, the aphids had started laying eggs and just making their own home. It was taken away from the actual plant by them eating the root and its way of taking in the nutrients. So what we did was sprayed for them with the second or third week, but it still did not kill them. We initially sprayed for these mites, but in the end result, we ended up having aphids inside the roots. So really, they were there the whole time, and it was a never-ending problem with that. Third, within the system, the roots would get so thick towards the end of harvest that water was not able to get through the actual channel. It would clog, and the roots would just ball up. So that prevented the water flow from going through the roots and into the recirculating system that pumps the water constantly. So there was either just too much air being pumped through, which as well can kill the water pump, because if there's not enough water and it's just air, the pump will just fail. And we did have that happen once. We did solve that quickly though. Leaks. This was a big issue. The leaks would start out from the sides, as you can see the top right, the sides of the channels, like within the middle, and then on the bottom left, as you can see where those metal pipes are, the water would go and flow underneath those metal pipes, and then drip throughout the day constantly. So we figured out that. So we took out the first plant, as you can see right there. Every single plant right there, we took that out so the water was able to flow just completely through it. And we took the channels apart a little bit, so there wasn't all the pressure in there. So yeah, basically the water would leak so much to the point it would drain all the 50-gallon tanks, and it would end up making us refill at each tank with 20 gallons. So I mean, it was a constant going back every day and a half just to make sure it wasn't empty. The next thing was the treatments varied. This was our day-to-day thing. So from about week 3 had begun, we started to see that the treatment 1 was not growing in sync with the other ones. As you can see in the bottom left, the first part of the picture is treatment 1, and then the back side is treatment 2 and 3. And as you can tell, those two look way thicker and way healthier than the ones in the front. So we did day to day. I mean, we checked the temperature, making sure those were right. But the temperatures we had were, in the first one was 86 degrees. So that was scorching them. We feel like that was scorching and preventing them from growing that well. And the other two the second one was 77 degrees Fahrenheit, and the last one was 68 degrees Fahrenheit, which was the best one out of all 3 treatments. We had 3-gallon tanks that were, no, that's right. We had 3 tanks that were 50 gallons. I mean, 2 to 4 days we'd have to go clean, as I said, add substrate, as I said. And we used this pump system that had our substrate and the tomato and cucumber substrate. So all I'd have to do is flip a couple switches and turn on the water, and it'd flow through the hose. And once I got done, I'd take it apart, turn those back off, and turn the ones on for the tomatoes and cucumbers on, so I wouldn't mess up their experiment as well. But for the future of this, for improvements, I would say having larger channels, because the roots are just they just grow way too thick. And as well, I would think having bigger recirculating tanks, because you never know if they're gonna leak, or how much you want to have just in case of the leak, because it could be make or break for your harvest. As well, the cooler temps for this, ranging from 68 to 77 degrees, was the best of growing the 4 strains of kale. But 68 was the best. I feel like that should be the one used for years to come. what was as well, figuring out which strains of kale grew better, and we found that the Red Russian and arugula was the best for the NFT system, and the others look good, but they did not look as edible and as good to the eye as Red Russian and arugula.